Usually when we talk about a medical diagnosis, there's this expectation of absolute precision.
Oh, sure. Yeah.
Like, we treat the human body almost like an engineering project. You break your arm, the X-ray shows a jagged white line on a black background, and the doctor just points to it.
Right. It's binary. Broken or not broken.
Exactly. It's clean, it's binary, and it's visible.
And, I mean, we really crave that visibility in medicine. A clear image or a single definitive number on a chart, it provides a lot of comfort. It makes the body feel, categorized and manageable.
But looking through the massive stack of research articles, linguistic textbooks, and the personal notes you sent over for this deep dive, it struck me that our most common, universally experienced diagnostic test is, well, it's nothing like that X-ray.
Not at all.
It doesn't require a multi-million-dollar imaging machine. It's entirely free. You produce it every single day. And historically, the diagnostic landscape surrounding it is incredibly murky.
It is literally the original muddy water of medicine. We are, of course, talking about urine. Before we had blood panels or MRIs or genetic sequencing, this fluid was the ultimate and, frankly, the only real diagnostic window we had into the living body.
Which brings us to our mission for today. We are taking a deep dive into a tiny linguistic building block that serves as the master key to this entire world. It's the suffix -uria, urea.
Derived from the ancient Greek word aryan, meaning urine.
Exactly. And this little string of letters might seem like basic medical jargon to you listening, but as your notes pointed out, it is a hidden window into centuries of medical history.
Oh, absolutely. Bizarre biochemical breakthroughs and some truly wild scientific debates.
It's fascinating. So let's start at the foundation. When you or I see urea at the end of a medical term on a chart, what is it actually telling us? Because the textbook definition makes it look like a simple compound word.
Right. At first glance, it is. The strictest definition, what linguists call the compositional sense, is that urea signals a specific substance is present in the urine. Usually something that shouldn't be there or is there in an abnormally high amount. The root word tells you the substance, and the suffix tells you where it was found.
Right. So if you have glucose in the urine, you get glycosuria.
Exactly.
Blood in the urine is hematuria. Protein is proteinuria. Name the substance, add the suffix, and you have your diagnosis.
That is how it works for those specific conditions. But the reason linguists and medical terminologists actually argue over this suffix today is because it refuses to stay in that neat little compositional box.
Really?
Yeah. Over the centuries, it's been recruited to do triple duty. It doesn't just describe what is dissolved in the fluid.
Wait, what else could it possibly describe?
Volume and frequency, for one. There is an entire second category of urea words that have nothing to do with chemistry. Take polyuria, which just means producing an excessive volume of urine, or polyuria, which is dangerously reduced output.
Oh, I see.
And anuria means the kidneys have stopped producing it almost entirely. Plus you have nocturia, which just means waking up multiple times at night to go.
Wait, but you could have polyuria, like too much volume, with perfectly healthy, normal chemistry, right?
Absolutely. You could just be drinking massive amounts of water. The chemistry might be pristine, but the term still uses the exact same suffix as a serious chemical imbalance.
That seems confusing. And you mentioned triple duty, so there's a third category.
Yeah, the third duty folds in the physical sensory experience of urinating. The main example here is dysuria, which simply means painful or difficult urination. So now the suffix is describing a localized physical sensation.
Not the chemical makeup and not the volume.
Right.
Okay, let me pause here, because structurally, this is bizarre. It's like if a mechanic had a single word ending for car problems that applied equally to finding rust in the engine oil, driving 500 miles in a single day, and getting a cramp in your calf while pressing the gas pedal.
That is a brilliant analogy.
Why on earth is medical terminology so messy?
It's a great question, and it's the exact reason there is a quiet war between terminology purists and medical pragmatists.
Purists versus pragmatists.
Yeah. The linguists and purists look at this and say, we need to clean this up. They want urea locked down exclusively for the chemical substances.
The rust in the oil, to use the analogy.
Exactly. They argue that using the same root for volume and pain just muddies the diagnostic waters.
But the medical field hasn't changed it. So the pragmatists must be winning. Why keep it so broad?
Because the pragmatists are rooted in how medicine was historically practiced. Before modern laboratories, doctors didn't have the luxury of separating a patient's pain from the volume of fluid or the color of it.
It was all just one thing.
It was all one holistic diagnostic event. To a doctor hundreds of years ago, the physical pain, the frequency, and the fluid itself were indivisible.
Wow.
Modern medicine kept the terminology because it reflects that history of observation.
And that pragmatic approach, looking at the whole picture,
Yeah.
it didn't just appear overnight. Your notes specifically brought us to the Greeks for this.
Yes, to the Hippocratic corpus in the 5th and 4th centuries BCE. This era birthed the practice of uroscopy.
Uroscopy.
Right. Ancient Greek physicians conceptualized the body as being governed by 4 humors. And they understood that urine was a biological exhaust of those humors.
Okay.
It was a mirror reflecting the internal state of the body, so they would collect it in a specific type of glass flask called a metula.
Right, I've seen pictures of those.
And they would meticulously examine the color, the viscosity, the cloudiness, and the sediment that settled at the bottom.
But they couldn't run it through a mass spectrometer. They only had their own 5 senses.
Oh.
So they're looking at it, they're probably smelling it.
Smelling it was vital. The odor of decay or sweetness told them a lot. And famously, they tasted it.
I knew we were going to end up here.
Yep.
The taste testing, I mean, it takes a very specific kind of dedication to look at biological waste and think, you know what, I should take a sip of this for science.
It sounds totally repulsive to us now, but it was rigorous sensory data collection for the time. By the medieval and early modern periods, this practice, often called water casting, was the absolute gold standard of medicine. Physicians were actually nicknamed piss prophets. If you look closely at European art from the 15th or 16th century, the visual shorthand for a doctor isn't a stethoscope.
What is it?
It's a scholar holding up a round glass urine flask to the light.
That's amazing. So who were the standout piss prophets? Like, who actually moved the needle on these uria diagnoses using just their senses?
A major turning point happened in 1674 with an English physician named Thomas Willis.
Okay, what did he do?
He was treating patients suffering from a mysterious wasting disease where they produced massive volumes of urine. Willis applied the sensory method. He literally tasted his patient's urine.
What did he find?
He documented that it was wonderfully sweet,
Oh, what?
as if it had been infused with honey or sugar. Because he tasted that sweetness, he appended the Latin word mellitus, meaning honeyed, to the condition.
To distinguish it from other diseases that just caused high volume.
Exactly. And that specific taste test is the direct ancestor of the diagnosis we now know as diabetes mellitus.
Which is wild.
And the sweet substance he tasted. That gave us the term glycosuria.
That is incredible. He used his own palate as a rudimentary chemical assay to define a disease we are still battling globally today. Though I have to point out from the research you sent over, Willis wasn't the first human to notice this.
Far from it. Centuries earlier, the ancient Indian physician Sushruta observed the exact same phenomenon.
But he didn't drink it.
No, instead of drinking it himself, he noticed that certain patients' urine strongly attracted ants and insects. He called it honey urine.
Using ants as your lab technicians, I mean, that is objectively brilliant and significantly more sanitary than Willis's method.
Much more sanitary.
So Willis finds sugar by tasting. But not everything the body excretes has a taste, right?
Right.
If a kidney is leaking something flavorless, how did early doctors catch that?
That brings us to another pioneer, Richard Bright, in 1827. Bright was studying kidney disease, specifically in patients with severe systemic swelling. They called it dropsy back then.
Dropsy.
He knew there was something wrong with their urine, but tasting it wasn't the answer. Instead, he subjected it to heat. He would take a patient's urine sample in a metal spoon and boil it over a candle flame.
Boiling it in a spoon. Again, the resourcefulness.
Yeah.
But what does boiling actually achieve there?
It reveals hidden proteins.
Mhm.
Normally, proteins in your blood are folded up into complex little 3D structures.
Okay.
When you expose them to intense heat, those structures unravel. They denature.
Like cookie and egg.
Exactly. The unraveled proteins tangle into each other and solidify. It's the exact same chemical mechanism as dropping a clear, liquid egg white into a hot frying pan. It instantly turns opaque and solid.
Wow.
Bright saw this white coagulation in the heated spoon and realized he was looking at albumin, a blood protein.
So he just cooked the urine until the hidden protein cooked like an egg white, and that gave us the concept of proteinuria.
Exactly. He proved that the kidneys of these patients were leaking vital blood proteins out of the body.
Which is a perfect pivot, actually, because all of this history,
Mm-hmm.
the tasting, the boiling, the ants, it's all observing the output.
Right.
But how do these substances actually get into the urine in the first place? I mean, to really understand urea, we have to understand what is physically happening inside the body.
We do. We have to look at the mechanical filter itself, the kidney.
Okay.
The kidney's core function is a constant balancing act. It filters your blood, reabsorbs the molecules your body needs to survive, and excretes the excess waste and water as urine.
Makes sense.
When you see an abnormal urea, like sugar or protein, it usually means one of two major systemic failures has occurred.
What are they?
Either the kidney's reabsorption capacity has been totally overwhelmed by too much of a substance in the blood, or the physical filters themselves are broken.
Let me see if I can translate this mechanism for everyone listening. Let's imagine your bloodstream is an exclusive, high-end nightclub.
I love this. Okay.
The kidneys are the bouncers working the front door. Their job is to keep the VIPs, like proteins and blood cells, safely inside the club while kicking the garbage out the back door into the alley.
Which is the urine.
Right. So let's take that analogy a step further. What happens when blood sugar spikes?
Well, glucose is normally allowed in the club.
Okay, but if someone eats a massive amount of sugar, suddenly there are way too many rowdy glucose molecules showing up. The bouncers have a limit, right? A threshold.
Exactly. In medical terms, it's the renal threshold for glucose, usually around 180 to 200 milligrams per deciliter of blood.
Right. So the blood sugar hits 200. The bouncers are completely overwhelmed. They throw their hands up. The velvet rope drops, and the excess sugar just spills right past them, out the back door, and into the alley.
The result is glycosuria. The filter isn't broken, it's just overwhelmed by volume.
That makes so much sense.
But contrast that with proteinuria. Proteins are large, vital molecules. In your analogy, they are the ultra VIPs.
They should never be kicked out into the alley.
Never. If you find large proteins in the urine, it doesn't mean there's too much protein in the blood. It means the physical barrier in the kidney, a microscopic structure called the glomerulus, is actually injured.
Oh, so the bouncer isn't overwhelmed.
No, the bouncer has been knocked unconscious, and the VIPs are just wandering out the back door.
That makes the distinction so much clearer.
Yeah.
A threshold issue versus structural damage.
Yeah.
So urea is essentially the forensic evidence of what slipped past the bouncers.
Yes, and paying attention to that specific forensic evidence led to what I consider one of the most profound breakthroughs in the history of science.
Really? When was this?
It happened in 1902 with a physician named Sir Archibald Garrod. He was examining a patient whose urine had a very alarming property.
Alarming how?
When it was excreted, it looked completely normal, but if it was left sitting out, exposed to the air, it turned pitch black.
Pitch black. That sounds like something out of a horror movie. Why would it turn black?
It's a condition called alkaptonuria. The black color is the result of oxidation. The urine contains a specific chemical called homogentisic acid.
Right.
When that acid hits the oxygen in the air, a chemical reaction occurs that turns it dark. It was famously noticed in infants when their cloth diapers would turn black after being soiled and sitting in the laundry pile.
So Garrod is looking at these black diapers. What did he actually figure out from that?
Well, he spent years studying families with this condition. And in 1908, he delivered a series of lectures where he laid out a revolutionary idea.
Okay.
He proved this wasn't a bacterial infection and it wasn't a kidney injury. He deduced that the patients were missing a single specific enzyme whose job was to break down a specific amino acid.
Oh, wow.
Because the enzyme was missing, the half-broken down acid just built up in the blood until it spilled over the threshold into the urine. He coined this an inborn error of metabolism.
Wait, 1908. That is decades before Watson and Crick. Decades before we even knew what DNA looked like.
Exactly. Garrod deduced the concept of genetic inheritance of metabolic diseases using a urea condition as his primary evidence.
That is staggering.
He didn't have a genome sequencer. He observed black urine oxidizing in the air, mapped the family tree, and essentially founded the entire field of biochemical genetics using sheer logic.
He deduced an invisible missing enzyme from a dirty diaper.
He did. And that framework of looking for missing metabolic enzymes in the urine paved the way for another massive milestone. In 1934, a Norwegian doctor named Asbjørn Felling was examining two children with severe developmental delays.
Okay.
Following Garrod's logic, Felling decided to test their urine. He added a chemical compound called ferric chloride to the samples. Instead of a normal reaction, the urine flashed a brilliant, unnatural green.
Green this time. What did the green mean?
The green flash proved the presence of phenylpyruvic acid. Felling had just discovered phenylketonuria.
Better known as PKU.
Yes. These children were missing an enzyme to break down the amino acid phenylalanine. Because they couldn't break it down, it built up in their blood to levels that were highly toxic to their developing brain cells, causing severe neurological damage.
PKU. I mean, if you're a parent listening to this, you definitely know PKU. It's on all the hospital pamphlets.
It is, because Felling's discovery directly led to the work of Dr. Robert Guthrie in the 1960s. Guthrie realized that if you could catch this chemical imbalance right at birth, before the toxic buildup damaged the brain, you could save the child.
Just by changing their diet, right?
Simply by feeding them a special diet free of phenylene. So he developed the famous newborn heel prick test. It is one of the greatest public health triumphs of the 20th century.
That's amazing.
Countless lives have been saved from permanent brain damage, and it all started with a green chemical reaction in a urine sample.
It really highlights how many rare diseases are defined by what ends up in this fluid. I mean, the sources you share even mention maple syrup urine disease, which sounds completely made up.
I know, but it's a very serious metabolic condition.
Right.
Its formal medical name is branched-chain ketoaciduria, so it officially ends in uria. But the colloquial name persists because the missing enzyme causes amino acids to build up that literally make the infant's urine smell like sweet maple syrup.
Just like the sensory observations of the ancient piss prophet.
Exactly. Full circle.
Okay, so the diagnostic power of urea is undeniable. But as with any forensic evidence, it has to be interpreted correctly. What happens when doctors or historians misread these signs?
Oh, it can be problematic.
Because taking one symptom and running with it can be dangerous.
It can completely hijack a narrative. And one of the most famous historical examples of this involves King George III of England.
The monarch who lost the American colonies, famously known as the Mad King.
Right. He suffered from severe bouts of mental illness, hallucinations, paranoia.
That was the accepted history for a long time.
Yes. But in the late 1960s, two psychiatrists, McAlpine and Hunter, combed through the king's historical medical records. They found notes from his physicians mentioning that during his episodes, the king occasionally produced dark, discolored urine.
Just from that one note.
Based heavily on that one detail, they published a blockbuster paper claiming the king wasn't psychiatric at all. They claimed he suffered from porphyria, a rare genetic blood disorder that causes nervous system flare-ups and dark urine.
So they retroactively diagnosed the man who had been dead for over a century based on a note about urine color.
They did. And for decades, the porphyria theory became widely accepted. It was taught in medical schools and featured in movies.
Wow.
But more recently, modern medical historians and neurologists like Timothy Peters and Peter Gerard's research teams pushed back hard.
And what was their counterargument? Because we want to make sure we're just laying out both sides of this debate here.
Yeah, of course. They pointed out the danger of retrospective diagnosis. They analyzed the king's surviving handwritten letters using modern linguistic software, and the frantic, repetitive syntax perfectly matched the manic phase of bipolar disorder.
Okay, but what about the dark urine?
Well, they suggested it was likely caused by a medicine the king's doctors were giving him, a plant called gentian, which is actually known to turn urine dark.
So we have a massive historical cold case, the porphyria camp on one side, and the bipolar camp on the other.
Exactly.
It's a perfect warning about how a single reported urea finding can derail an entire diagnosis if you don't look at the whole patient. Which actually brings me to a very practical point.
The hypochondriac angle.
You guessed it.
Yeah.
If you're listening to this and you just got an alert on your medical portal that your annual physical labs are back,
Right.
you open the PDF and it says in red letters that you have proteinuria or glycosuria,
Yeah.
your immediate thought is that your kidneys bouncers are permanently fired and you have a chronic disease.
Panic sets in.
Yeah. Is a urea always a permanent diagnosis?
That is a crucial question, and the answer is a definitive no. The public often assumes any urea finding means irreversible disease, but these conditions can be entirely transient and completely benign.
The bouncers might just be having a bad day.
Basically, yes.
So what causes a transient leak?
Let's take proteinuria. You can spill protein into your urine simply because you did a heavy, exhausting workout the day before.
Really?
Or maybe you had a high fever from the flu. There's even a condition called orthostatic proteinuria, where teenagers literally leak protein just from standing upright for too long, and it completely resolves when they lie down.
That's crazy.
The physical stress temporarily alters the pressure in the kidneys. A little protein slips by, but the system recovers.
And what about sugar? Can glycosuria be transient?
Yes, very commonly during pregnancy.
Oh, okay.
The increased blood flow and hormonal changes naturally lower the kidney's threshold for glucose. Pregnant women will often spill sugar into their urine, even with perfectly healthy blood sugar levels.
Wow, so the bouncers just lower the velvet rope a bit.
Exactly. This is exactly why a clinician will never diagnose you based on a single abnormal test. They will always have you come back on a different day, well-rested and hydrated, to see if the condition persists.
Okay, that is a huge relief. So listeners, don't panic at the first lab results.
Exactly.
Before we wrap up, I want to ask about related terminology. Are there false friends? Like words that look like they belong in this family but don't?
The most common false friend is enuresis.
Enuresis, that's the clinical term for bedwetting, correct?
Yes. It sounds like it should be heavily related to uria, but linguistically it derives from a completely different Greek root meaning to void.
Oh, I see.
It's completely unrelated to the compositional suffix we've been discussing. However, uria does have a very close, true partner in medical terminology.
What's that?
The yin to its yang, and that is the suffix emia,
Like anemia.
or hypoglycemia.
Exactly. Anemia refers to the concentration of a substance in the blood. Diagnostically, uria and emia work together to tell the whole story of the body's metabolism.
They often describe the exact same substance, just from opposite sides of the biological filter.
So if we go back to our analogies, if emia is the massive traffic jam building up on the highway, urea is all the cars desperately spilling off the exit ramp.
That is a brilliant way to visualize it.
Yeah.
Glycemia is the sugar stuck in the traffic jam in the blood. Glycosuria is that same sugar flying off the exit ramp into the urine.
Okay, I see it.
Uremia, which is a very dangerous condition, is toxic waste backing up in the blood traffic jam because the kidneys have failed and the exit ramp is closed.
Wow.
You need both concepts to understand the flow of the human machine.
It is incredible how elegant that system is when you step back and look at it. We've traced this tiny suffix on quite a journey today.
We really have.
From Hippocrates trying to balance the humors in ancient Greece to Thomas Willis trusting his taste buds over his gag reflex.
Brave man.
Very. To Archibald Garrod founding the science of metabolic genetics just by looking at a dark stain on a diaper.
And it hasn't lost its relevance.
Not at all.
Even with all our modern technology, urine remains medicine's cheapest, most accessible, and most revealing diagnostic material. It powers the over-the-counter pregnancy tests we buy at the pharmacy.
Right.
It's how obstetricians routinely screen for preeclampsia. It's the foundation of every sports physical and drug test.
It all traces back to the diagnostic logic locked inside those four little letters.
It really is the ultimate biological window. But looking forward, it leaves us with something fascinating to ponder.
Oh, what's that?
We've gone from tasting urine in glass flasks to sequencing the proteins inside it, but we still largely test it only when we feel sick.
True.
Think about the future of this. Engineers are currently developing AI-powered smart toilets for our homes.
Wait, smart toilets?
Yeah, that passively run spectroscopic urea diagnostics every single morning. They'll track our metabolic trends before symptoms ever even appear.
Wow. A daily lab report from your bathroom.
Exactly. And on a larger scale, we are seeing the rise of wastewater epidemiology, where cities test the municipal sewage.
Oh, like for COVID.
Exactly, essentially tracking the macrouria of an entire population to predict viral outbreaks weeks before patients ever show up at a clinic.
That is wild to think about.
It makes you wonder, as we develop these continuous passive tests, what profound biological secrets, what warnings of disease are we currently just flushing away every single day simply because we haven't been looking closely enough?
The muddy waters of diagnosis still waiting to reveal their secrets. If you're listening, the next time you look at a lab report, you'll never see that little suffix the same way again.